comfortably seated in the fertility clinic with Vivaldi playing softly in the background. You and your partner are brought coffee and a folder. Inside the folder is an embryo menu. Each embryo has a description, something like this: embryo 78 male, no serious early onset diseases, but a carrier for phenylketonuria, a metabolic malfunction that can cause behavioral and mental disorders. Carriers just have one copy of the gene, so don't get the condition themselves. Higher than average risk of type 2 diabetes and colon cancer, lower than average risk of asthma and autism, dark eyes, light brown hair, male pattern baldness, 40% chance of coming in the top half in SAT tests. There are 200 of these embryos to choose from, all made by in vitro fertilization, IVF, from you and your partner's eggs and sperm. So, over to you. Which will you choose? If there's any kind of future for designer babies, it might look something like this. It's a long way from the image conjured up when artificial conception, and perhaps even artificial gestation, were first mooted as a serious scientific possibility. Inspired by predictions about the future of reproductive technology by the biologists J.B.S. Haldane and Julian Huxley in the 1920s, Huxley's brother Aldous wrote a satirical novel about it. That book was, of course, Brave New World, published in 1932. Set in the year 2540, it describes a society whose population is grown in vats in an impersonal central hatchery, graded into 5 tiers of different intelligence by chemical treatment of the embryos. There are no parents as such, families are considered obscene. Instead, the gestating fetuses and babies are tended by workers in white overalls, their hands gloved with a pale corpse-colored rubber, under white dead lights. Brave New World has become the inevitable reference point for all media discussion of new advances in reproductive technology. Whether it's Newsweek reporting in 1978 on the birth of Louise Brown, the first test tube baby, the inaccurate phrase speaks volumes, as a cry round the Brave New World, or the New York Times announcing the Brave New World of three-parent IVF in 2014, the message is that we are heading towards Huxley's hatchery, with its racks of tailor-made babies in their numbered test tubes. The specter of a harsh, impersonal and authoritarian dystopia always looms in these discussions of reproductive control and selection. Novelist Kazuo Ishiguro, whose 2005 novel, Never Let Me Go, described children produced and reared as organ donors, last month warned that thanks to advances in gene editing, we are coming close to the point where we can, objectively in some sense, create people who are superior to others. But the prospect of genetic portraits of IVF embryos paints a rather different picture. If it happens at all, the aim will be not to engineer societies but to attract consumers. Should we allow that? Even if we do, would a list of dozens or even hundreds of embryos with diverse yet sketchy genetic endowments be of any use to anyone? I don't think we are going to see Superman or a split in the species anytime soon, because we just don't know enough. Henry Greeley, bioethicist. The shadow of Frankenstein's monster haunted the fraught discussion of IVF in the 1970s and 80s, and the misleading term, three-parent baby, to refer to embryos made by the technique of mitochondrial transfer, moving healthy versions of the energy-generating cell compartments called mitochondria from a donor cell to an egg with faulty, potentially fatal versions, insinuates that there must be something unnatural about the procedure. Every new advance puts a fresh spark of life into Huxley's monstrous vision. Ishiguro's dire forecast was spurred by the gene editing method called CRISPR-Cas9, developed in 2012, which uses natural enzymes to target and snip genes with pinpoint accuracy. Thanks to CRISPR-Cas9, it seems likely that gene therapies, eliminating mutant genes that cause some severe, mostly very rare diseases, might finally bear fruit, if they can be shown to be safe for human use. Clinical trials are now underway. But modified babies. CRISPR-Cas9 has already been used to genetically modify non-viable human embryos in China, to see if it is possible in principle. The results were mixed. And Kathy Nyakin of the Francis Crick Institute in the UK has been granted a license by the Human Fertilization and Embryology Authority, HFEA, to use CRISPR-Cas9 on embryos a few days old to find out more about problems in these early stages of development that can lead to miscarriage and other reproductive problems. Most countries have not yet legislated on genetic modification in human reproduction, but if those that have, all have banned it. The idea of using CRISPR-Cas9 for human reproduction is largely rejected in principle by the medical research community. A team of scientists warned in Nature less than 2 years ago that genetic manipulation of the germline, sperm and egg cells by methods like CRISPR-Cas9, even if focused initially on improving health, could start us down a path towards non-therapeutic genetic enhancement. Besides, there seems to be little need for gene editing and reproduction. It would be a difficult, expensive and uncertain way to achieve what can mostly be achieved already in other ways, particularly by just selecting an embryo that has or lacks the gene in question. Almost everything you can accomplish by gene editing, you can accomplish by embryo selection, says bioethicist Henry Greeley of Stanford University in California. Because of unknown